in 3.3, it's just another state again. It's asking you to remember that DNA is replicated before meiosis begins. So in this image, if we can, I wish there was like a before, because this is kind of like the after. So this chromosome here, its homologous pair is this blue one here. So let's pretend this is like chromosome number 1. So there was one pink one and one blue chromosome number 1, one from your mom, one from your dad. And same over here, if this is chromosome number 2, we have this is homologous to this one. So what this is saying is that during interphase, we make copies of all of these. And this is still considered one chromosome, but it consists of sister chromatids, which is some confusing terminology, but we will practice it, I promise. So 3.34 is just talking about the phases here, and I'm not going to spend a lot of time on just walking through this and saying exactly what this is. I'm going to point out some of the key features. So in these images, you see these two little dots here. These two little dots are eventually going to be the centrioles, and centrioles are where spindle fibers come from. And spindle fibers are these strings, these protein strings that are going to grab onto these middle parts of the chromosomes called centromeres. But the important thing in prophase 1, and we'll practice this, I promise, but crossing over is like one of the main reasons we have genetic variation. So I don't know if you can see it here, but if we look at this chromosome, it ends up crossing over with its homologous pair, right? Remember, we made copies of these before. And what happens is the alleles are switched. So if dad gave you blue eyes and mom gave you brown eyes, now dad has brown eyes on his chromosome that he gave you, and blue eyes, I kind of forgot what I said, but blue eyes is on the mom's chromosome. So alleles are exchanged. That's a very important part of meiosis. And the next phase is metaphase. Metaphase is when these centrioles that we talked about before reach out and grab onto these centromeres. Also, the chromosomes, these homologous pairs, line up in the middle. That's how I remember metaphase, middle. In the next stage, it's called anaphase. And anaphase is the stage where chromosomes begin to move apart. So these spindle fibers are getting shorter and shorter and pulling them in. This is when our chromosome number reduces. And a lot of times this is confusing, but this is the time we have the number because, I'm not sure what happened, but that's okay. So what we've done is we've taken, because this is one chromosome, and I know the terminology is weird, but this is one, even though it's made of 2 sticks of DNA, it's one chromosome. So the homologous pairs have been separated. So this is the time where we go from 2N to N. That's the point that happens. After anaphase, there's telophase, or sometimes it's called telophase. So telophase is this next one. In telophase, what happens is the nucleus reforms. That's the big thing. And we are no longer diploid, as we mentioned. And then these cells will divide into 2. And then we do the whole thing over again. So there's an interphase, but there's no replication. So we keep that reduced number of chromosomes. Prophase 2 is where the nucleus begins to break down. That way the spindle fibers can grab onto them. There is no crossing over in prophase 2, only in prophase 1. Then we have metaphase again. So in metaphase, just like the other one, they line up in the middle. That's how I remember. The centrioles contract. And then we also have anaphase 2. The chromosomes will move away from each other. And then we'll have telophase 2, where we end up with haploid nuclei again.